like I mentioned, it's a lab and a mouse. If you open this up, you see a whole bunch of little guys in there and at a little electronic stations doing all kinds of different things and sending answers back to the computer. And what does that lab and the mouse do? We target brain function, and the way we target it is by vibrating the fingertips. When you vibrate fingertips, you know, the sense of touch is very unique compared to other senses. One is that it's a really high fidelity system. That means you can send very clean signals to two places in the brain that are side by side. If I stimulate these two fingers, I'm going to activate two places in the brain that are side by side, right in the middle of the brain. And you know, we're going to activate these places, and then we're going to ask questions. We just ask really hard questions, like which one came first? Which one lasted longer? Which one was bigger? So in other words, I'm being sarcastic. Questions are really, really easy. Now the stimuli we deliver aren't necessarily, are somewhat complex, but the questions we answer are really easy. So all of you have probably taken an eye exam. Eye exam's pretty easy conceptually, right? Starts out easy, and then it gets harder and harder and harder. We do the same thing with the brain gauge, except what we're doing is we're targeting brain function. We're probing brain function with the brain gauge. We're sending stimuli to different parts of the brain, and we're asking questions about it. And those questions we get answers about a whole bunch of different mechanisms, a bunch of different fundamental building blocks. So things like, you know, and this is a list of those things: lateral inhibition, information processing speed, feed-forward inhibition. These are all different mechanistic fundamental building blocks. And so how do we do that? And basically we try to take that idea, and we answer those questions. When we answer those questions, what we do is we ask those questions, and we get numbers for all those fancy terms, all those fancy mechanisms, and then we come up with something a lot more simple, where we just say, okay, you have speed, accuracy, some executive function, timing perception, and we're looking at, you know, here are the different measures. We convert those mechanistic numbers into something that's really easy to understand, easy to follow. And we try to make it so that you can sort of look at brain function and decide, well, this seems easy. How many people can tell that day 1 is worse than day 14? Would you rather have scores that look like day 14 or day 1?
Day 14,
right? We try to make it intuitive. So we all green, good, you know? Not all green, not good. Well, this was day 1 post-concussion for somebody. You know, somebody got whacked in the head, they get tested, and they say, well, yeah, you're not ready to play again. Still not ready to play. Okay, you might be ready to play again.